Good afternoon, class. I welcome you again to Metric Hub. Today we are going to talk about genetics and inheritance terminologies, key terms. There are certain key words that we are supposed to define so that when we are going to do our chapter of genetics and inheritance, all things will be easy and flowing. I'm going to ensure that I explain and give the examples of each of the terms that are going to be used there. The first term that we're going to talk about is monohybrid cross. Monohybrid cross, which is a mating or a cross between two homozygous parents that differ in their alleles for one gene. That results in a monohybrid offspring. So in terms of this, or with regard to this one, monohybrid cross, the parents are homozygous and they are differing in one of their alleles for a gene. So what we need to understand is that these organisms, we want to understand, and I would wish that you get this understanding from the reference that was given by Gregor Mendel. That is our reference that we are going to use in this explanation. So the two parents are homozygous and they differ in their alleles for one gene. So when we say they are homozygous, what do we mean? These two parents, we are saying they are purebreds and they are differing in their allele for one gene. So let us just quickly work on these terminologies. So in this case, we are saying it is a tall plant, a tall pear plant that is crossing or that is crossed against a dwarf, which is a short pear plant. So what we are saying is that these two parents, they are what? They are homozygous. In this case, we are saying their genes, their alleles are the same. They have the same alleles, as to mean to say that the tall plant is homozygous, meaning to say that it is homozygous dominant, and also the short plant is also homozygous, which is homozygous recessive. So in this case, I just want you to see that this is also called homozygous. What? This is also called homozygous. It's called homozygous. This one we are saying is called homozygous. This is homozygous dominant. Homozygous dominant. And this one is saying, we say it is homozygous. It's homozygous recessive. They are in a case or in a way, both of them, they are homozygous, meaning to say that the two alleles that are there, they are the same, they have the same strength. So we mean to say they are homozygous. That's what it is. And then our next term is monohybrid, which is an organism that is heterozygous for one gene. Our next term is what is monohybrid. Monohybrid, an organism that is heterozygous for only one gene. It has two different alleles for that gene. Heterozygous, as you can see, or the understanding that you should just have the literal meaning of hetero, meaning to say different. So in this case, in terms of the genotype representation, this organism, it is having a dominant and a recessive allele. So what it means is that the organism is heterozygous for one gene. It has two different alleles for the gene. The two different alleles for the gene, in this case again, is that we are having a recessive allele and a dominant allele that are being joined together there. We are saying we have got the dominant and the recessive allele that are being illustrated or that are being shown in this context. This is a recessive allele that is shown, and also we have got this which is also, we can just illustrate to say it is also showing a dominant allele there. Dominant allele, the capital letter T there is dominant allele. So what is happening is that in genetics, I'm sure that you understand that we always use letters, which are either capital letters and small letters, to represent characteristics, where the dominant allele is represented by a capital letter and the recessive allele is given by a small letter. We are saying a characteristic or a gene that is the same, we cannot use different letters for its contrast in terms of the dominant and the recessive. What it means is that a dominant allele is always given by a capital letter, and then the contrast of a dominant allele, which is a recessive, is given by a small letter of that same alphabetic letter. Hence, we say that there is a capital letter T and a small letter T representing dominant and recessive alleles there. What is genotype? Genotype, the genotype of an organism. What do we mean by the genotype of an organism? The genotype of an organism, if you can see there, we are saying it is the genetic makeup of an organism. It is the genetic makeup. The genetic makeup, it is in the blood. If you can remember very well, or if you can see very well, what we mean is that the genotype cannot be seen by eyes. It's something that we can see by the letters that are used to represent the situation or the scenario that is being in question. So we use the genetic makeup as to say it is represented by letters. Letters are given that represent the alleles in the organism, which we are saying, for example, in this case, we are saying the genotype can be given for a tall plant. We are saying it is given as what? As homozygous T dominant is the genotype, or we can also say, or we can as well say it is homozygous T recessive. It is still representing the genotype. So the genotype is always supposed to be given as letters. Letters of alphabet are the ones that represents the genotype. Please take note of that. Letters of alphabet are used to represent the genotype. Letters of alphabet. We are saying genotype is given by letters of alphabet. That needs to be taken note of, and it must be well understood that the genotype we always use, it is given as letters of alphabet. Letters of alphabet represents the genotype. In any scenario, whether you are in monohybrid, crossy, dihybrid, crossy, complete, incomplete dominance, whenever you are asked to give the genotype, understand that it is the genetic makeup that you are looking at. You are looking at the organism's genetic makeup, the genes that are making that thing, which is given in the blood, that is giving a certain representation when we go to look at the phenotype. The phenotype of an organism, the phenotype. The phenotype, we are saying it, we need to understand to say phenotype is the outward appearance. It refers to the visible expression of the genotype. In other words, we can give it as to say, like in this case, is the interpretation of the genotype, which we can say it is either to say tall, or it is either to say short. So what we are saying is that the phenotype, it is the interpretation of the genotype. Phenotype, we can say is the interpretation of the genotype. We can say it is the visible expression of the genotype. It is the visible expression of the genotype. Is the visible expression it is the visible expression of the genotype. So what we are saying is that the meaning that is given to say what is those letters representing, we read to understand the meaning of the letters, then we can give the phenotype. The phenotype is supposed to be giving a description. It is supposed to give a descriptive term that we can say it is tall, it is short, it is black, it is dwarf, it is what you call any other colors or any other terms that we can use that can describe a situation. We call that the physical appearance. The physical appearance of an organism, which is the phenotype, Our next term is homozygous. Our next term is homozygous. What do we mean by homozygous? Homozygous, we are saying individual that has two of the same alleles. Remember to say our chromosomes are always homozygous in nature, and then we are saying they are two of the same alleles of a gene. So in this case, is in terms of representation, in terms of the phenotype, if we give, in terms of the genotype, we can give this phenotype as to say, the homozygous as to say that there is the representation of an individual that is two of the same alleles, which we can say it can be tau, homozygous tau dominant, or we can have homozygous tau recessive. Homozygous short. So homozygous, it can be either dominant or it can be recessive. Then we are saying it can, both scenarios, they are representing homozygous. And then heterozygous, what do we mean by heterozygous? Heterozygous individual that has two different alleles of a gene. The two different alleles of a gene, they are also represented in the genotype, which we are given to say we have got a dominant and a recessive allele. So whenever we are given a heterozygous individual, in terms of the genotype, we are saying it is a dominant and a recessive allele that is given or that is used there in terms of the genotype when we look at the heterozygous. Heterozygous under monohybrid crossing, we also have it as to say it also sometimes results, it results in what is called complete dominance. It can also result in what is called complete dominance. What we call complete dominance. So complete dominance, if you can see there, what we are saying is that the dominant allele will define the outward appearance. It will define the physical appearance of an individual. The dominant allele will result into the meaning of the phenotype of the individual. Therefore, we say that we call that the meaning of this individual. We are saying its phenotype, the phenotype of this heterozygous will be what? Will be tau as a result of the dominant allele. That is in the heterozygous, which is in the heterozygous scenario there. So tau is equal to tau, which again, I would wish that you can as well understand that when we are into this representation or into this scenario of monohybrid crosses, we also understand that individuals or organisms that are heterozygous are equally the same as to homozygous. So we are saying homozygous dominant is equals to heterozygous is equals to tau if we are going to represent the phenotype. So they can only differ in the what in the genotype, but when we interpret the genotype, it can mean the same. To say homozygous dominant is equals to heterozygous dominant, it is equals to tau. That is why. And next term is a purebred. What is a purebred? An individual that is homozygous for a characteristic. A purebred is, according to Gregor Manteo defined that he crossed organisms that were purebreds. A tall pear plant was crossed. A tall pear plant was crossed with a short pear plant, and in this case, we are saying both of them are purebreds. They are purebreds, both of them. They are purebreds. They are producing, they are going to have individuals that are going to be a combination, or that are going to be a reflection of the dominant allele that is being expressed in the scenario, or that is being involved in each of the cases there. And then last term, but not least, is hybrid, a new type of a plant formed by cross-pollinating different varieties of the same plant. We are saying the plants are of the same type, but we cross-pollinate them. As to say, we are taking the different alleles of the different plants. Therefore, we cross them together. When we cross those together, the results, or they give us an individual that is what we call a hybrid. It is going to be a hybrid individual. And then we also have another term, which is the locus. Locus. What is locus? Locus is the position of a gene on a chromosome. Is another term that we should also look at. Locus. 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 What is it? What we call locus? Locus. We are saying it's a position of a gene on a chromosome. When you look at a chromosome, chromosomes, they carry genes. So it is the position, of a gene on a chromosome. On a chromosome. We call that locus. Position of a gene on a chromosome is called locus. We have another term which we can as well add there, or other terms we just need to add few, but not all of them. We can as well add another term, which is what is it? Allele. Allele. Which is a variation of a gene. Allele, we are saying variation of a gene. Variation of a gene. That is an allele. For example, we can talk about, let's say, we are talking about the gene for height. We have got short, we have got short, we have got tall and short. So tall and short, there are variations of the gene for height. Then we are saying that is an allele. I think these terms, they are going to help us in the definition and understanding of the calculation that we are going to do now. We want to go and talk about genetic crosses. We are going to talk about genetic crosses, genetic diagrams. We are going to talk about genetic diagrams as our next subtopic, subheading, genetic diagrams as our next subheading. genetic diagrams. Genetic diagrams. What are we talking about? What do we mean? What are we going to discuss here? Genetic diagrams, they are actually used to understand inheritance of characteristics in organisms. Organisms, they need to, we always understand, or we always want to worry, why do organisms produce young ones that are exactly or that looks alike to them? So genetic diagrams help us to understand how do offsprings obtain or inherit these characteristics from their parents. They also illustrate the genotypes and the phenotypes of the parents, which would result in the young ones that we are going to, or that will be produced. So in that case, we do have symbols that are used, as to say, in terms of generations. Generations can be generations are represented by P. They are represented by F, where P is representing the parental generation. Generations are represented. We are saying symbols are used. Symbols are used to represent. We have got symbols that are used to represent. Symbols are used to represent generations. Symbols are used in the diagrams that we are going to use there. We have got different symbols that are used. For example, I'm saying, let's say in this context, in this scenario of the genetic diagram that we are going to talk about here now, we have got what you call letters of alphabet that are used. So I'm going to just use a simple flow diagram that would show these symbols as they are being used. So we have got P, P1, P, it represents parental generation. It just represents the parents' generation. P represents parents' generation, which we are saying P1, we are referring to say it is the parental generation, which is the first parental generation. And then we also have F. So P, we can have P1 and P2, which of course are representing the different generations of parents. We also have F, which can be either F1 or F2 as well, also representing the filial generations. We are saying F1, if it is F1, it is representing first filial generation, saying F1, we are saying first filial generation. And of course, F2 is saying second, is representing also second filial generation. Second filial generation. There are generations there. We also have the alleles of a gene can be represented by capital letters and small letters. Alleles of a gene are represented. Alleles of a gene alleles are represented by letters of alphabet. They are represented by letters of alphabet, where we are saying the dominant allele is represented by a capital letter, and then the recessive allele is the small letter represents the recessive. The recessive allele is represented by a small letter, and then obviously, like we said, is representing the dominant the dominant allele as well. The dominant allele is represented by dominant by capital letters. So I would want us to just quickly have an example of the calculation that Gregor Mantel did when he was explaining the monohybrid crosses. We want to calculate the F1 generation, and then also the F1 generation as well as the F2 generation in this representation, or in this diagram, in this context. We want to talk about the F1 and the F2 generations. Gregor Mandel talked about breeding and crossing homozygous individuals that are pure breeds. So we are going to quickly do that. P1, we say P1, we start with the phenotype. The phenotype, we say that he talked about, in this case, we are talking about tau PA planting is being crossed with a shorty PA plant. Is being crossed with a short PA plant, and then we're saying that is the phenotype, and then we say genotype. I'm sure you still remember the terms. We say the genotype is the what? Is the genetic makeup. So in this case, we said these plants, they are pure breeds, and they are obviously pure breeds. We said that they are what? They are homozygous. They are homozygous dominant and homozygous recessive. And then, having said this, we are saying these plants, after we are showing the genetic diagrams. So meiosis must occur. Meiosis has to take place. When meiosis takes place, it gives us what we call gametes. And then the principle of meiosis is to divide or to halve the number of chromosomes in an organism. So we are saying, in the example that we are having here, we are going to have one T there. And then it is going to be crossed with also a small t, single t there, because meiosis is divided by 2. It divides by 2 these gametes. I mean, these are homologous cells there. So it divides by 2. So then we can have small one dominant T and one recessive T. Those are the gametes. And then similar again, we can give this example as in terms of the number of chromosomes in a human cell, where we say that is a full set of cells or a full set of chromosomes in a human cell, in a human body. We are saying it has got 46 chromosomes, but when it undergoes meiosis, it has to have 23, which is half the number of 46. So there we have got a double T dominant and double T recessive. So it gives us single T dominant and also single T recessive. Having that, we are supposed to have fertilization. Fertilization must occur. Fertilization, as you all understand, we said it is the fusion or the what? It is the chemical process that takes place by the fusion of the two gametes there, which we are going to have a heterozygous T there. Heterozygous T, again, let's not forget that this is also this is the genotype, because it's the genetic makeup. It is still our genotype that is given there. It is still our genotype. So what is the meaning of this genotype? We're saying the genotype must always give us the meaning of the phenotype. So the phenotype is the meaning of this genotype, the interpretation of the genotype. What is the meaning? The phenotype, we are saying, is the meaning, is the representation or the expression that is given from this, which is going to mean to say what? Which is going to mean to say tau. So our F1 of a monohybrid cross, it has got 100% tau. we can say 100% tall. They are 100% tall, as to say the dominant allele, which is giving us also in this case, it's a complete dominance that we are seeing because we have got the dominant allele that is giving the phenotype, as to say that these plants are all tall. So F1 is 100% tall of the dominant allele. It is the F1 generation of a monohybrid cross. So we want to also, as you can see, this is also similar, or it is also the same explanation that we have given there, to say complete dominance would always give a representation of the phenotype of the dominant allele. The dominant allele would give the meaning of what is being interpreted. So in this case, like we said there, this is a heterozygous combination, which we said that it would give us to say this is equals to, or the heterozygous combination that we have given there, to say heterozygous is complete dominance, which is equals to tau. The dominant allele is giving, the dominant allele is equals to tau, which is homozygous T. Dominant is equals to heterozygous T, which is equals to tau, the phenotype. That's what it is. That's what is happening there. So we also want to look at the F2 generation. The F2 generation. Let's quickly work out the F2 generation. The F2 generation, how do we work the F2 generation? F2 generation, we said we start also with the P, as we have seen there. We said P2. P2 represents the parents, the second generation of parents, which I have shown you here. The first P, we are saying P1 parents generation. So if it is P1, it is the first parents generation. And then if it is going to be P2, it means the second parents generation, which we also have our phenotype. So our phenotype in this scenario, again, it is a tall pear plant. Tall pear plant is crossed against a tall pear is crossed against a tall pear plant. And don't forget that we said that this F1 becomes the P2. F1, the offsprings becomes the P2. So F1 are the ones that we are taking to represent the P2 here that we are showing here. So now, what is the genotype here? The genotype is just well given there, which is the one that we are having here, to say heterozygous T. So we have got a heterozygous T plant, which is crossing against a heterozygous T plant there. And then meiosis must occur as well. Meiosis, so therefore we can have our what? Our gametes. So our gametes are going to be, we are going to have our dominant T, we are going to have our recessive T is crossing against our gamete, which is dominant T, as well as recessive T. And then having said that, we are saying they should be what? There should be fertilization. Fertilization must take place. Fertilization, we are saying, is the fusion of the gametes as they cross, or as they come against each other. So we have got this T dominant there and that T dominant there. We have got that TT. And we also have the first T again here. It must finish everything, which is also on the what? On the right. So we have got this T and then that recessive T, which is awesome to say that this T and then that recessive T there. And then we have got this T here, the dominant T, the dominant T here, it is crossing, the recessive T here is crossing with the dominant T there, and then we have got the heterozygous T there, and then we have got these recessive T's coming again on their own. The recessive T is coming across on their own there. So what we are having is that we have got a dominant, we have got these outcomes that are produced when gametes of F2 generation are crossed together. At the same time, we are still saying this is the genotype. It is the genotype. So this genotype, as it is like that, we want to give the meaning of the genotype. What does it mean? What is the meaning of this genotype? What does it mean? This genotype, we give it to say, we interpret it to say this is one, it is two, this is two, this is two. So we have got three two here as to one short. As you can see, this is short. We said that heterozygous T is equal to homozygous T, which is why we are saying it is what? It is 3 toe there, because we have got toe here, toe, 1 toe, second toe, third toe, and then we have got 3 toe as to one short. This, again, we have said that this is the outcome, which can as well be given as what? We are saying give we are saying give these answers. We are saying give these answers. Let me say give as percentage ratios. Give this as percentage ratios. We said that it is going to be chance factor, which is going to be saying chance factor of tau is 3 over total ratio 3 plus 1 over 4 times 100 over 1 is to 1 chance factor of short over total ratio 4 times our 100%. And then we are saying that would give us to say 3/4 of 100, it is 4 into 100. 4 into 100 is 25, and then 25 times 3, we have got 75. That is 75% as to also here, 4 into 100 is 25, and then as we get that, we get also 25 times 1 is 25, which is also percent. So the answer would therefore mean to say that we have got 75% to 75%. This is 75%. This is 75% to as to 25% short. 75% to as to 25% short. And I think I've given you this as a genetic flow diagram. Let me just quickly summarize this through a Punnett square, because most of you easily understand a Punnett square than a genetic diagram. So I can as well do the calculation through a Punnett square, which we are saying that a Punnett square can as well be used to calculate and represent chance factors in what? In genetic crosses. We can as well do through a Punnett square, which we can say this is going to be saying our toe, or saying our male parents 1, and then our toe female parents 1, mean parents 2, which are also going to be showing also through this side. So our genes, our gametes, in this side we have got our toe planting, which is heterozygous, and also we have got also the gamete here, which is our toe pair plant, which is heterozygous as well. So we are saying the T, this one, and the T, that one, they come together. We have that T, this T also the dominant T, and then hit small t there, we have it. We take this T here, which is that dominant T, and the the recessive T there. We take this small t there and that small t there. We have got this, and then again, if you can look at it again, this can as well give us to say, giving us equally the same answer, to say we have got equally the same answer of saying we have got 3 tau here. We have got 3 tau as to say this is tau, this is tau, this is tau. So we have got 3 tau as to 1 short, which is exactly the same answer that we got there to say that this 75% as to 25% also is the same answer that we got there to say 75% as to 25%. Having said this, we are saying this is our F2 generation. This is our F2 generation of a complete. This is our F2 of a monohybrid cross. So you can either use a Punnett square or you can use a genetic cross, a genetic flow diagram. A genetic flow diagram, it is also just easy, but I mean that sometimes students, they get confused or they lose it. But what you need to understand is that before you go to the Punnett square, you should always have the representation, because your markings, when the question comes, you are going to be asked such that you will be able to understand to say what is being crossed. So we are saying it is a tau-PA plant that is being crossed against a tau-PA plant, which you can see here now to say if you are going to use a Punnett square, you should start also from understanding that what are you crossing? You are crossing the tau-PA plant against a tau-PA plant there, which we are saying up to this level. When you come to this level, you can therefore divert and come to use what? You can therefore divert and come to use what? A Punnett square, because you are transforming, you are picking these gametes here, and then you come and insert them into the Punnett square that we have drawn here to say that these are heterozygous gametes against heterozygous gametes, and then they cross together there in order for you to have these chances that we have given there: 3 tau and 1 short, 75% tau and 25% short. I'm sure that you would get this well. Thank you very much for your time. And in our next presentation, we are going to be talking about codominance. Codominance is going to be our next presentation. I'm going to come and show it to you. Thank you very much. And I wish you all the best. Good day. Thank you.